Hello, everyone, and welcome to today's session, Automating NGS Workflows for Precision Medicine Oncology, CeGaT's Experience. I would like to start by introducing today's speakers. Dr. Elisabeth Matischneck is a Product Manager Oncology at CeGaT. She has a PhD in molecular biology from the Medical University of Vienna and several years of experience in translational oncology and clinical research, as well as research project management. Elisabeth joined CeGaT in 2019, and her role focuses on supporting customers conducting clinical trials from requests to data interpretation. I will be the second speaker. My name is Gabriela Bosa Moran, and I am the Scientific Content Manager at Hamilton Robotics. I have a PhD in molecular and cell biology and an MBA in Management of Technologies. I have more than 10 years of experience in scientific research, and my role at Hamilton is to create and to disseminate scientific content describing the use of Hamilton products in various applications. At the end of the webinar, Dr. Svetlana Konko will join us during the Q&A as our Hamilton automation expert. Svetlana has a PhD in radiation biology and more than 5 years of experience in automation of projects. Svetlana is a sales manager for the German and Austria sales team for Hamilton Customized Automation Solutions. Before we begin, I would like to mention some important aspects. This webinar is being recorded and will be available for download. We would love to hear from you, so please write your questions in the Q&A box that is at the bottom of your screen, and we will do our best to answer all your questions by the end of the webinar. If for some reason we're unable to do so, please feel free to contact us after the webinar, and you will find the contact and the email at the last slide of the webinar. For those who are joining us now, welcome. We will now begin. I will start just talking about the agenda. The agenda for today's webinar is as follows. So Elisabeth will first give an overview of CeGaT, describing all the systems and the workflows for NGS library preparation. She will then give an overview of the role of precision medicine oncology, followed by some case studies, and then she will finish with an outlook about NGS precision medicine oncology. I will then give an overview about Hamilton and our automation solutions for NGS, with a focus in oncology studies. And I will describe some of the reference that we have here for scientific content around this topic. We will finish then with a Q&A session. And as I previously said, please, you can start writing your questions as we go along in the Q&A box. I will now pass the presentation to Elisabeth.
Thank you, Gabriela, for the kind introduction, and welcome everybody. I'm glad you're here today with us. So I would also like to start with an introduction of our company, CeGaT. CeGaT is a medical diagnostics company which was founded in 2009 by Saskia and Dirk Biskup, and we are still privately owned. And here below you can see the building we are located in, which is in Tübingen in the south of Germany. A lot has changed since the company was founded a bit more than 10 years ago. It went from a few ambitious people with an idea to about 300 employees at the time now, and almost half of them are holding a MD or PhD degree. 5 additional very important employees of ours can't be seen in the picture because those are our 5 NovaSeqs, our sequencing systems. We are also using Hamilton liquid handling workstations. At the moment, we are using 6 of them. And we are also using other automated systems like the Kia Symphony, for example. What is it that drives us? What is the vision of CeGaT? Well, we want to be the partner of choice for research institutions and pharmaceutical companies around the globe. And we want everybody to see what we see, which is the manifold applications and the possibilities NGS has to offer. And we want to close the gap between, on the one hand, standardizing NGS services, but still being able to deliver the ideal solution for our customers' needs. The CeGaT has 2 business fields. One is genetic diagnostics, And here our aim is to identify the genetic cause of a disease, understand the background, and then at the end we deliver a full medical report for the referring MD. And then there's the second business field, research and pharma solution. That's, for example, where I work in. Here our customers are typically research institutions, but also pharmaceutical or biotech companies, which focus on basic or clinical research. Here the portfolio is a bit larger, and we don't deliver a full medical report, but the raw sequencing data and the result of bioinformatical analysis. Here a little overview on CeGaT's history. I already told you we were founded in 2009. And soon after that, we focused on introducing diagnostic panels, which means sequencing a certain set of genes, which is being linked to a certain disease. And then in 2011, a very important step for us, we were accredited according to ISO 15189. In 2014, we moved into our own building, which meanwhile is becoming too small already, so there will be a new one soon. In 2015, CeNATA was founded. That's a subsidiary of CeGaT, and CeNATA is performing NIPT tests, so non-invasive perinatal testing. And then in 2016, we became accredited from CAP and CLIA. And in 2017, it was CLIA. And then in 2018, another subsidiary was founded, the CeCAVA, which is focusing on neoantigen-specific tumor vaccination. As you can imagine, the development of sequencing technologies within this period, this 10 years, was massive. So we went from analyzing a few genes in single samples to massive parallel sequencing with a capacity of up to 1,000 whole exome samples per week at the moment. But still, we are a medical diagnostics company. That means we have to fulfill highest quality standards, and we have to assure that we deliver consistent quality for every customer. And we have implemented several strategies for meeting those demands, while at the same time increasing our capacity. One is constant sample tracking. So once a sample arrives here at CeGaT, it receives a barcode and is tracked throughout the whole process. Another one are our certifications for according to ISO and from CAP CLIA, which are very important also to receive feedback on what you are doing and to keep your standards high. Another one is the quality management system, which assures that everybody is receiving regular trainings. We have internal audits and things like that. And for us, one of the key pillars for our strategy for increasing the throughput, but still being able to meet those quality demands is automation. And in the next slide, I'm going to tell you why. So I'm going to talk about automation here at CeGaT now. And actually, I briefly want to mention that those slides were prepared by my colleague Isabel Hake, which of course wanted to join us today, but she's not able to. So I'm very sorry if I might not be able to answer every automation related question in very detail, but I'll try my best here. So I already told you that the number of samples we need to process in the lab has increased dramatically over the years. At the same time, the turnaround time of the project shouldn't get any longer, but rather shorter. And therefore, we had to increase throughput. However, if you have to process a higher number of samples, that also increases the risk for mix-ups or other mistakes. And you also have to assure reproducibility, meaning that the results should be exactly the same, independent if person A or person B did the experiment. In order to take all of that into account, we have decided to automate our library preparation protocol. So the library preparation is basically the key element of NGS. And we use Hamilton Systems for that. So one main advantage is that the robot handling system can deal with a lot more samples at the same time than a person could. So we receive higher throughput. At the same time, we can document which sample has been processed in which way, so we receive higher traceability. And the robotic system generates reproducible results, no matter if person A or person B pushed the button for starting the run. So we have a high reproducibility. So for us, automation brought a lot of different benefits. In general, one could say that NGS and automation is a combination which is quite tricky, and it's not that easy because NGS is a relatively new field. It's developing fast. That means that the protocols need to be adapted, new protocols need to be implemented. And if you automate something, then this protocol is set, and it could mean a lot of effort for adjusting those protocols. Therefore, one of the requirements for automation for us was that it's a system which can be programmed by our employees themselves, and which allows us to react in short time. So we are flexible and we can adapt to necessary changes in the protocol very easily. So that's, for example, what the software of Hamilton looks like. And it's really rather simple. It can be handled even if you're not experienced in that area, because you don't have to perform classical programming based on codes. You can simply click on the different modules available and then combine them, put them together in a script, and then you define certain variables for your different sets that you have. So it's a rather intuitive system. So we've got easy programming and flexibility for continuous script development covered. Another requirement for us was flexibility of the platform deck. So the flexibility of the Hamilton DAX allows us to only use those components which are required for the specific protocol we are using right now. Those components can vary between the different processes, and carrier can be exchanged easily, and thus we can optimize the deck in a way that there's very little user interaction necessary. For example, if a certain process doesn't require the use of tubes, we can remove them, and that remove the tube carrier, and then we end up with more space for other things like tip carrier, for example. And there are many different consumables available which can be used also during the same protocol. And that way, we don't have to waste any reagents or samples. If, for example, for a certain reagent, we have a large amount, then we can use a throw, which results in faster turnaround times. But if we have very little material, if it's really precious material like the samples, then we would use tubes instead, and we have less dead volume, so we're really flexible. And since the Hamilton is also equipped with modules like an on-deck cycler or a cool carrier, we can also perform temperature-sensitive steps right directly on the deck, and there's no need to transfer a sample back and forth onto other machines, but we can all do it on the same machine, which is also a great benefit for us. So here is an example of how a deck could look like, and some of the carriers that can be used, which give you quite a lot of flexibility, like the plate carriers, tube carrier, or tip carriers. Our NGS workflow can be divided into three main steps. The first one is that we isolate nucleic acids, and that can, for example, be done using the Quire Symphony. Then the second part is that we select the respective NGS library preparation protocol and perform the library preparation, and then that can be supported by the Hamilton Microlab Star, for example. And once the libraries are generated, then we sequence them, usually on our NovaSeqs. But not all of our library preparation protocols are performed in an automated fashion. Here we decide, based on the frequency we use the protocol, whether we want to automate the protocol or not. And if we decide we want to automate it, then our approach is quite straightforward. We implement the protocol that we usually would do manually on the Hamilton. So the protocol is exactly the same, then we would do it manually. And then we run tests first with water, then with reagents, but without actual sample. And if we see that all that steps are performed correctly, then we run actual samples. And then we compare the results of the samples which were processed automatically with the Hamilton and with those results generated manually. And then usually there are some adjustments necessary, and we do that until we end up with a protocol which leads us to the same or even better results than with the manual approach. So that's what the protocols look like for us. For example, the library preparation protocol is divided into 5 different steps. First, the actual library preparation. So that would be ligating adapters, pre-capture PCR amplification, purification. And then the next step is pre-capture pooling. So since the libraries have an index already, we can pool different samples together already at that step before performing the hybridization, because that saves reagents and thus costs. And then during hybridization, the libraries bind to baits, so we can enrich for the specific sequences of interest. For example, if you perform whole exome sequencing, then you would enrich for the exome sequencing and get rid of the intronic sequences. So that's already basically step number 4. You capture using baits, and then you wash to separate between wanted and unwanted sequences. Then you perform a QC again, and you can finally pool the finished libraries and you can sequence them. And those 5 different steps are programmed as separate scripts for us because it gives us a maximum flexibility. But besides reproducibility and standardization, of course, one main goal of automation is that you want to increase throughput and you want to be faster. So what difference does it really make? And that's an example of our whole exome sequencing protocol, library preparation protocol. And I think the most obvious difference for us is that we can process a lot more samples at the same time. Manually, this would be 32 samples, and automated, it's 96 samples at the same time. And on a daily basis, that really makes a huge difference. And of course, also the hands-on time is reduced a lot. And also the other steps, the other steps are really reduced. So for us, automation really brought a lot of advantages. So next, I would like to talk about precision medicine, because we already heard about automation in NGS, but the topic of the webinar is automating NGS workflows for precision medicine oncology. So what does NGS and automation have to do with precision medicine? And in order to answer that question, I would first like to answer the question, what is precision medicine? Because it's a term you hear very frequently nowadays. What do people really mean? And I found this definition in a publication. And it says that precision medicine is a treatment paradigm that takes the molecular and cellular features of a tumor, as well as those of its microenvironment and additional traits of the individual, such as genetics and lifestyle, into account to create a tailor-made treatment. So in other words, this treatment concept acknowledges the fact that everybody is different. So in the past, we believed that a person simply has lung cancer. And then maybe based on histology, we divided between non-small cell lung cancer and small cell lung cancer. And in fact, those two already have a completely different molecular therapeutic profile. But now we have methods available like NGS, and they're evolved so fast that we had the opportunity to characterize those cancer cells even on a molecular level and not just based on histology. And now there's a molecular characterization showed that we can divide them further into subgroups, for example, based on the presence of EGFR mutations or PI3 kinase amplifications or mutations. And we end up with a lot of different subtypes, not just non-small cell lung cancer and small cell lung cancer, if we also take those molecular findings into account. But does it really matter? Is there a difference, or is it just a nice to know? So the tumor has a EGFR mutation, and so what? Is there a consequence behind it? And yes, it does matter. First of all, because those tumor types, those subtypes often behave very differently. One could be much more aggressive than the other, and that could have an effect on treatment regimen, for example. So maybe those patients are treated for a longer period, or those patients have a different prognosis. But also very importantly, those molecular findings also have an effect on the treatment. In the past decade, the field of oncology has witnessed substantial changes in the way patients with cancer are managed. So the field came to realize that the one-size-fits-all approach is not suitable for such a complex disease like cancer, with every genetic background, every immune system and lifestyle being different in every patient. So one-size-fits-all is clearly not the right approach for such a disease. But now we can sequence tumors, we can identify driver mutations, so the mutations that underlie tumor growth. And that knowledge has led to the development of a lot of targeted therapies, which are based on those driver mutations. And in this figure, for example, you can see all of the different targeted approaches which have been approved or which are still under development for adenocarcinoma of the lung, for example. And you can see that it's actually quite a lot. And even, for example, for non-small cell lung cancer, the prognosis of those patients has really changed dramatically since we have discovered those drivers like EGFR mutations or ALK rearrangements, for example. And in the upper figure, you can see which type of lung cancer treatments have been approved over the last decades. There's traditional chemotherapy in orange, and you can see that in the last decades, it was mainly targeted therapies and also some immunotherapy approaches. But of course, that's not just limited to lung cancer, and it's also not just some short-lived trend. We've seen targeted therapies on the rise for quite some time now. And it's also one of the major reasons why the outcome of many cancer patients has improved a lot over the last decade. You can see here, for example, this part and this part reflects the targeted small molecules and targeted biologics which have been approved in the last years. And currently, there's more than 90% of the treatments in late-stage oncology pipelines are targeted small molecules or biologics and not standard chemotherapy anymore. So the number of druggable tumor-specific molecular aberrations has grown substantially in the past decade, and it was associated with a significant survival benefit for those patients. But precision medicine for us starts with precision diagnostics. So our vision is that patients receive precision diagnostics, so not only histology, but already the identification of molecular changes in the tumor from the beginning on. And that this combination is taken into account when selecting the treatment. So that could help patients to receive the best treatment right away and not just as second or third line treatment. And it could also expand treatment options for rare or aggressive cancer. And this tumor profiling can also help to identify driver mutations for which targeted therapy is approved, but maybe for a different cancer type. But still, the patients could benefit from that knowledge if we perform molecular characterization of the tumor. So therefore, this molecular characterization really has become a fundamental to drive therapeutic decisions. And our vision would be that that the current practice is adapted accordingly. However, if you want to give the patient some targeted treatment, you first have to identify the tumor's molecular changes. So that means first you have to do a genetic analysis. And for many people, I think a genetic analysis means you want to find out, is mutation Y present in gene X? So you're looking for one specific single nucleotide variant. That's how it was handled for a long time. But looking into clinical routine, what would that mean? It would mean that you resect the tumor, you prepare tumor sections, and then you check whether your patient has the specific mutation you are looking for, or if he doesn't. So taking the same lung cancer example, you would start for looking for the mutation with the highest probability. For example, you would prepare tumor sections, analyze, and you want to see, does the tumor have a KRAS mutation, for example? And if you're lucky, it does, so you can start treatment. But maybe you're not, and then you have to prepare more sections. Then you look for the next mutation, which has the next highest probability. For example, then you look for EGFR, or you look for ALK. And you continue until you end up with either finding a mutation or you run out of material. And access to tumor material is often a big problem in treating cancer patients. But there's more than single nucleotide variants. You can also look at insertions and deletions. You can look at copy number variations or translocations, for example, and you can do it in parallel. And more than that, you can not only look at all of those changes in a single gene, but you can also expand it and look at more genes in parallel. So you can analyze a gene panel, for example, or even perform more comprehensive genomic profiling and look at whole exome or whole genome at the same time. And that's possible with NGS. And that's why NGS technology have increasingly substituted for conventional techniques, because it allows to simultaneously not only look at single nucleotide variations, but also, for example, on copy number variations or translocations, and in multiple genes in parallel. So it provides a more efficient, cost and tissue-saving tumor analysis as compared to single gene biomarker analysis. For example, at CeGaT, we also use a lot of different panels, and for cancer patients, one we use quite frequently is our somatic tumor panel. And that involves the analysis of more than 700 cancer-associated genes. And if you, for example, need a high tumor coverage and you want to analyze it in a cost-effective manner, then you would go for this panel approach. Often, however, it is advisable to even perform more comprehensive analysis, like whole exome sequencing. And I will later talk about a case report where you can see all of the different insights which can be gained just from whole exome sequencing. So going back to our example, if we use NGS and not single gene testing, but rather analyze a larger panel or whole exome, we can check all of those genes in parallel and find the right treatment choice right away. And we don't have to guess and don't have to use precious time. We lose, but we can identify the most suitable treatment right away. And that's also where automation again comes into play. So replacing single gene testing with NGS and analyzing large panels or even whole exome is only possible if this can be done in a cost-effective way, and if it can be done in a clinically acceptable time frame, because those are therapy-relevant results and they are time-critical, of course. So analyzing large panels, whole exome, instead of single genes can only be done if you manage that high throughput while keeping the high quality. So if you want to meet all of those demands, then for us, one of the solutions was automation. So using those automated NGS approaches for precision diagnostics, we think we are able to introduce a shift of costs from therapy to diagnostics. In the last minutes, I've already talked about how NGS can generate molecular information for selecting the treatment, but there are also a lot more possibilities on how NGS can support precision medicine. For example, it can be used to monitor the disease and identify progression, Or you can also perform retrospective analysis and then find out why a patient responded to therapy or not. You can identify new combinational therapies, or you can use it for uncovering novel targets for therapies, identify resistant mechanisms. You can also identify patients which are eligible for a certain clinical trial, or off-label use of a treatment. As mentioned before, the treatment is intended for use, for example, in a certain cancer type, but the patient has another cancer type, but the same molecular characteristics, then you could use it as off-label use, for example, and also for identifying prognostic markers. And I found that statement in a publication, and I think it's a quite nice summary of what NGS can do in terms of precision medicine. So the genomic characterization of cancer is leading to the identification and use of better diagnostic, prognostic, and predictive biomarkers and more effective management. So next, I would like to talk about some case studies which show you practical examples of how NGS can be used for precision medicine in oncology. So in this first study that was conducted in cooperation with the Department of Dermatology in the University Hospital of Tübingen, here we assessed the use of TMB, tumor mutational burden, and circulating tumor DNA as a biomarker for immunotherapy in patients with metastatic melanoma. And I think maybe not all of you might be familiar with cfDNA, with liquid biopsies, and tumor mutational burden, and therefore I will just briefly explain what those terms mean. So cfDNA stands for cell-free DNA. It's also often referred to as circulating cell-free DNA because it's present in the blood, but also in urine or other body fluids. But usually when you analyze it, you would typically analyze it from plasma. And a few characteristics of cfDNA, it is quite fragmented because it mainly originates from dying cells undergoing apoptosis or necrosis. It has a very short half-life of just a few hours. And now the most interesting part for us, some part of cfDNA derives from dying tumor cells, and it means that it carries tumor-associated genetic and epigenetic alterations, and therefore we can use it as a tumor-specific biomarker. So this part, the tumor-specific part, is then referred to as circulating tumor DNA, ctDNA. And this analysis of cfDNA or ctDNA actually has a lot of advantages compared to common tissue analysis. First of all, quite obvious, it's non-invasive, the sample collection. You just draw blood instead of you having to perform biopsies. And that also allows you to collect samples repeatedly. And therefore, you can, for example, monitor the tumor evolution. You can monitor treatment, which wouldn't be possible if you would stick to tumor tissue analysis. And also, cfDNA is a representation of the whole tumor heterogeneity, because if you take a biopsy, then it's just a small part of the tumor that you are selecting and that you're analyzing, but you're not seeing the whole picture. And it also has the advantage, due to the short half-life, that it's always a current picture of the disease. It's tumor-specific, and it's a cost-effective way of analyzing tumor-specific, the tumor. And the other biomarker, which is used in this study and also in the next case report I'm talking about, is tumor mutational burden. So the unit of TMB, tumor mutational burden, is mutations per megabase. So it refers to the number of gene mutations which are present in the patient's tumor, but not in the healthy tissue. So we really are looking at coding somatic mutations. and that's a pan-cancer marker. So you need to analyze either a large panel or the whole exome in order to calculate the TMB. So that's only possible using NGS. So in this case study that I mentioned before, we analyzed TMB and liquid biopsies, and the patients had received immunotherapy. So while the patients had received therapy, we conducted repetitive liquid biopsy analysis. So we collected blood and analyzed the plasma for cfDNA or ctDNA every 3 to 4 weeks. So based on the tumor tissue, we identified certain driver mutations and we tracked them longitudinal during the course of the treatment. And here you can see a typical profile that you end up with. So you would see a certain mutation in the blood, in the cfDNA of the patient before you start the treatment. Then during the treatment, for example, the patient responds and you can't detect the mutation anymore. But then again, after a while, you might see the level of ctDNA rise again, and that's a sign that the patient isn't responding anymore. And most of the time, then also at the later time point, imaging can confirm that there's progressive disease. And as I told you, we also analyzed the tumor mutational burden. And here we did that by analyzing using our somatic tumor panel, so that more than 700 gene panel I told you before. And you can see that we divided the patient into 3 different groups. That patients that showed a complete response, those that showed a partial response, and those that progressed during treatment. And there's a large difference between those groups in the TMB values of the tumors of those patients. And you can see that those patients with the best response had the highest TMB, meaning the tumors had the most mutations. And at first, that might sound like a contradiction. Why are a lot of mutations? Why is that a good thing? But you have to consider that if there are a lot of mutations in a cancer cell, then the cell looks very different from a normal healthy cell. And that makes it easier for the immune system to recognize that something is wrong with the cell. And so what we learned from this study was that TMB was significantly higher in responders than in non-responders. And this high TMB was even associated with a survival benefit compared to the patients who had TMB low or intermediate tumors. But we also assessed the cfDNA, as I told you before. And also the cfDNA and ctDNA was associated with response to the immunotherapy and even also with overall survival. So if we saw a decrease in cfDNA concentrations of more than 50%, or if ctDNA was undetectable at the first follow-up, which was only 3 weeks after treatment initiation, those patients did a lot better. They responded to treatment and they had an improved overall survival. And so it was possible to estimate the response to this treatment very early on, even 3 weeks after treatment initiation. So you don't have to waste time on a non-effective treatment anymore. So that's also one example where comprehensive diagnostics led to the identification of patients which are suitable for a certain treatment, for example, immunotherapy. And at the same time, you identify mutations then, which you can monitor during the treatment, so you can find out very early on if this patient is going to respond to the therapy or not. Now, the second case study, and that's a case report that I find very interesting. It's the story of a patient who was diagnosed with urothelial carcinoma. So the patient was referred to us after recurrence during palliative chemotherapy in 2016. So it was recurrence in palliative chemotherapy. So there was basically nothing to do for this patient. The doctors couldn't help him anymore, but he was 55 and was referred to us. But they ran out of treatment options. We then performed sequencing of the tumor tissue using whole exome sequencing. And again, we evaluated the tumor mutational burden of this tumor. And the TMB was quite high. And as we saw before, patients with a high TMB are more likely to respond to immunotherapy. So the patient then received immunotherapy and he responded really well. However, the treatment had to be discontinued due to side effects at a certain point. But then we continued with another treatment, which was multipeptide vaccination, so cancer vaccination. That means that the patient had received a really personalized vaccine of different peptides based on the neoantigens that we have identified in the patient's tumor. So that's really the most personalized treatment option there is. So it was a vaccination really based on the mutations and the changes in the patient's tumor. And during the vaccination, we monitored the immune response. We looked at certain T cell fractions, and we saw that there was a strong increase in the CD4 and CD8 fraction. So the patient was responding, the immune system was reacting. And furthermore, we also analyzed liquid biopsy samples, so cfDNA samples during the treatment. And you can see the results here, all of those dots. So during the treatment, while the patient was responding, we couldn't detect any ctDNA, any mutations in the plasma of this patient. But then the vaccination was stopped as well, but still we continued to monitor the patient. So not monitoring the therapy anymore, but monitoring the disease. And at a certain point, we saw we were able to detect tumor-specific DNA in the plasma of the patient. And we could confirm it also at the next time point. And we talked to the treating physician, and he performed then an MRI scan, which proved tumor recurrence. So the tumor recurred, and that's what we saw in the liquid biopsy samples already. And therefore, the patient started immunotherapy again. And again, he responded. The ctDNA level dropped again, and the patient did well and is still doing well now in 2021. So I think there are several lessons to learn from this impressive case report. One is the treatment success of checkpoint inhibitors, which by that time were still off-label use, and also with the personalized neoantigen peptide vaccination. But also we saw how successful liquid biopsy monitoring was, not only for during the therapy, but also for disease monitoring. And it could have even had a benefit on the patient's outcome, because most likely the immunotherapy wouldn't have started as early if we hadn't indicated based on the liquid biopsy analysis that there was a recurrence of the tumor. And we also saw the importance of comprehensive genomic profiling. That's not something you could do without NGS or with single gene testing. So based on this case report, we can see it was we performed whole exome sequencing, one analysis. There were so many different insights you can gain from this one analysis. So it's again an example of personalized diagnostics followed by personalized treatment and monitoring. Because based from this whole exome sequencing, we learned that the patient had a high TMB, which means it was a candidate for immunotherapy, which turned out to be true. We were also able to identify driver mutations, which we then monitored during the treatment and also afterwards. So we found out about the recurrence of the tumor. Also, based on whole exome sequencing, we identified neoantigens, and those were the basis for creating the personalized vaccine that the patient was treated with. And we also performed HLA typing to select those peptides with the highest affinity. So a lot of different insights from whole exome sequencing. And now the third and last example of the use of NGS for personalized medicine. And that's not a study we were involved in, unfortunately, but it was done from the National Cancer Institute. And they wanted to find out what is special about exceptional responders. So they defined exceptional responders as patients that showed a partial or complete response to a treatment that usually would only be effective in less than 10% of similar patients. So they took 110 of those exceptional responders and they characterized them on a genomic level. So they were looking for DNA mutations, the RNA expression profile, DNA copy number alterations, and so on. And also the immune cells of the tumor microenvironment. And in more than 23% of those patients, they found actual molecular features that could potentially explain why the patients did so well and why they responded so well to the specific treatment, although all of the other patients didn't. And they could categorize it. There were 4 main categories which could explain why the patients did so well. One was that there were different genetic changes which co-occurred in the patient's tumor. So there was an effect called synthetic lethality present in those patients. Another one was the body's ability to repair DNA damage. Another one was intracellular signaling. And also very important, the immune system's response to immune cells. So the study showed that genomic characterizations of cancer can uncover genetic alterations that may contribute to the unexpected and long-lasting responses to treatment. So the study certainly gave very interesting insights which can be exploited therapeutically, but they can also provide several testable hypotheses regarding the oncogenic mechanisms that may influence the response to cancer therapy. So it's also a very important study which might give a lot of clues for the future. And with that, I would like to end with an outlook on what we think that the future of precision medicine will look like, or what we wish it will look like according to us. So what we would like to see in the future is more integrated approaches. So we think precision medicine could look something like that. Cancer is a very complex disease. So for us, it makes sense to not only look at the tumor properties, but also take the immune status and the microbiome of the patient into account. So you would, for example, of course, analyze the tumor properties like mutational burden. You would use cfDNA for monitoring, but you would also look at, for example, T-cell receptor diversity and the gut microbiome, which plays an important role in all different kinds of diseases. Also, it's, for example, linked to the response to immunotherapy. And what we think, what are the next steps towards achieving the goal of precision medicine, especially with regards to NGS, is on the one hand, as I already mentioned, our integrated approaches, more flexible and interventional clinical trials, and then a change in paradigm away from single gene testing towards comprehensive genomic profiling, because you just can get a lot of more information out of this analysis. It's more, you have more flexibility, it's more cost effective. You can go back retrospectively to the data to see, to analyze the data, because you might not know what you're looking for by the time you do the analysis, and that allows you to go back. Also, one very important aspect in the future, we think, is support for clinicians, molecular tumor boards, because nowadays you get so much information, also with NGS data being, giving you a lot of information. We don't think it can be handled by just one person, so it really requires molecular tumor boards, and they will become increasingly important. Another one is standardization, because that allows reproducibility and to compare the results of different studies, and high quality standards of the labs where those NGS tests are performed, because that's the basis for establishing trust in NGS, trust from the patients, from clinicians, from pathologists, that this data can really help them. Yeah, and with that, I would like to thank you for listening, and I'm happy to answer your questions later.
Thank you, Elizabeth. And now we will pass through a brief, just a brief explanation of the solutions that Hamilton can offer for NGS. So Hamilton, to give you an overview and explanation about our company, it's a private group founded in 1947. And today Hamilton has two headquarters, one in Reno, Nevada, and one in Bonaduz, Switzerland. We have a total of 5 manufacturing sites. And in the places, so we have one in Franklin, one in Romania, and another one also in Switzerland. All in all, the company right now employs more than 3,000 people around the world, and this includes 7 subsidiaries throughout Europe and China. Hamilton is divided in 5 independent business units. Hamilton Robotics is specialized in the manufacturing of liquid handling instruments, both for OEM and for direct sales. And this webinar has been focused in the products and the services of Hamilton Robotics. Hamilton Storage manufactures ultra-low temperature automated sample management systems and small systems for capping and decapping. Hamilton Process Analytics manufactures measurement devices that includes pH sensors, dissolved oxygen, and conductivity. Hamilton Laboratory Analytics manufactures syringes for use in various applications. And finally, Hamilton Medical manufactures ventilators for people in critical care. Hamilton Robotics has 4 platforms for automated liquid handling. So we have the Microlab Vantage, the Microlab Star line, the Microlab Nimbus line, and the Microlab Prep. The systems that Lisa was talking about and that they use in CeGaT are based on our Microlab Starline. Our solutions for NGS. So we currently offer 2 as-ready workstations for NGS, the NGS Star and the clinical NGS Starlet. So as-ready workstations are predefined deck layouts that have been already programmed to perform qualified workflows. That means that end customers already equipped with everything they need in terms of hardware and in terms of software to perform their experiments. So in terms of, we have here the Hamilton platform, the classification, the throughput, and the available methods. The NGS Star, it's based on a Star platform. It is a research use only instrument, and it can process up to 90 samples at the same time. The clinical NGS Starlet is based on a Starlet platform, which is a bit smaller than the Star, but it's also part of the Star line. It is an IBD compliant platform and is optimized to process up to 24 samples at a time. And here show then some of the methods that have already been or available or qualified on our systems and are specifically for the study of precision medicine studies. So here we have Illumina's TrueSight Tumor 170, TrueSight Oncology 500. We also have Cancer Hotspot Panel B2, Focus Panel, Comprehensive Panel B3. So if you're interested to know any particular method, if you already have it available, please feel free to contact us. So as we all know, ERAD will dramatically increase the regulatory oversight of all diagnostic products for 2022. And we will go from what we call now IBD Directive to IBD Regulations. And this is expected to have a significant impact in the way clinical laboratories work in the future. And this is particularly important for laboratories doing NGS analysis for precision medicine studies. This is given the fact that the majority develop and validate their own assays. And this is what we call LDTs, homebrew assays, or in-house tests. Yet, for 2022, only LDTs for which an equivalent CE-IBD does not exist, meaning that no commercial kit is available, will be permitted for use. Hamilton offers directly or through its partners IBD compliance system like the clinical NGS Starlet, And we also fully validated IBD systems like the very sick NIPT Microlab Star here at the bottom. This is a system that was co-developed and it's sold by Illumina. And it includes the Hamilton liquid handler with the Illumina developed scripts, so the software that is run on Venus, as well as the necessary reagents and the consumables. The very sick NIPT Microlab detects genome-wide fetal genomic abnormalities of maternal peripheral blood. So I would like to highlight, without going into much detail, but I would like to highlight some of the tools and modules available in our Microlab systems. Specifically, these are also available in the systems that they have in CeGaT. Most of them are available in our 4 platforms. So first, we have our platforms can be integrated with single independent channels of 1 milliliter or 5 milliliters, or they can also be integrated, can be integrated with 96 or 384 probe heads. And they all can with our core technology for reliable pipetting using air displacement. We offer various tools for moving objects on deck, and here we see one of them that is available on the STAR platform, the Core Gripper. The Autoload is a module that is placed at the entrance of the instrument, and it automatically loads the carriers and reads the barcodes, as well as the barcodes of the tube and on the microplates. Our platform can also be equipped with Ineco cold plate air cool, or we call it by short CPACs, and they allow the heating and cooling of the samples from 4 to 110. And the CPACs are usually used for cooling of sensitive materials, such as reagents, and in some cases, as well as samples. The HSS, or the Hamilton Heater Shaker, is used for shaking and heating microplates in various SBS formats. The heating can go up to 105 Celsius. And when used in conjunction with the magnet for NGS protocols, it is usually used for the cleanup steps, as well as the capture and wash in all the parts that have library enrichment steps. Finally, here we have the ODTC, or on-deck thermal cycler, which is used for incubations between 4 and 99 degrees. The ODTC is commonly used for all enzymatic reactions, which include the DNA fragmentation, the end repair, the A tailing, the ligation, as well as the PCR amplification. So again, this is an overview of some of the models that we have on platform. Besides our solutions for automation of NGS library prep, we also offer solutions for many other steps of the diagnostic workflow. So here, this is a general workflow for molecular diagnostics, from sample collection to sequencing or to PCR. In dark blue are the steps for which we offer dedicated Hamilton assay workstations. In white are customized solutions, and in light blue, we have solutions with Hamilton storage. So for our assay workstations, we have for aliquoting samples, for fractionating blood, for doing the extraction of DNA or RNA, the ones we discussed about NGS library prep, and also for doing the setup of the qPCR. For customized solutions, we can also offer for the lysing the fluid, for the quality control, for setting up the plates before doing the quality control, also for reformatting the plates in order to fit the instruments used for quality control, and also for pulling the libraries. Together with Hamilton Storage, as I said, we also provide solutions for sample storage at -20 and also at -80 at different sizes depending on the requirements. Finally, I would like to invite you to take a look at the content that we have recently developed around the topic of NGS in precision medicine oncology. So first, we have an ebook which talks about the history of precision medicine oncology and how NGS plays a central role in it, why automation is important, and the solutions that we can offer. We also have a case study with CeGaT, with Elizabeth and her team, where we explain more in detail the automation of the workflows that they use for TMB and MSI analysis. We have 2 videos recorded at the customer sites showing how the instruments are being used in the lab. One video is with the Munich Leukemia Laboratory in Germany, and the other one is with the Clinical Genomics Laboratory of the Moderna University Hospital in Italy. Finally, this webinar will also be available on demand after today, so you will get this email. You might also be interested in two other of our ebooks in the area of genomics. So the one about precision medicine oncology, we also have NGS in diagnosis of rare diseases. And we have a third one in the molecular diagnosis of infectious diseases, where we talk about all a range of solutions for genomics. With this, I want to thank you, and I want to move now to the Q&A session. I know we are arriving now at 5, so I understand for those who need to leave now, but we do have a couple of questions, and I would like to then take a moment to answer them. You also had the opportunity to send your questions before. Now, Svetlana is going to join us to answer all the questions related to our Hamilton automation platforms. I will start now with the Q&A. So one of the questions that I have here is probably for Elizabeth. Why is your library preparation protocol divided in 5 different scripts?
So in theory, you could also use one script for everything, but we prefer to divide it because it gives us more flexibility. If we need to implement certain changes, then we can just implement it in the certain script and we don't need to test everything. And we can also stop after each step. For example, the hybridization takes 16 hours. And this time we use the step is performed off-tech. So in this time, we can use the Hamilton system for something else. And that's, for example, that kind of flexibility, which is easier if you separate the scripts and not just use one.
Thank you, Elizabeth. Another question is probably for both of you. So it's, do you need special programming skills or IT skills for writing the scripts in the Hamilton instruments? Maybe we start with Svetlana.
Sure, yeah. I think I would just basically also refer to what Elizabeth has actually already said, that no, you don't really need programming skills. You can work with the drag and drop menu for specific steps like pipette, move, and so on, and then adjust it to your needs. So you can set up a method as you would like to. However, to be very honest, is of course that I would like to point out that also what Elizabeth said in the very beginning, that an NGS protocol is not trivial. So it has its complexity. And if you imagine to set up a workflow, a manual workflow of 2 days, then you can also imagine that it will take a while. So, but you can do it without programming. Real code programming knowledge. And we offer some software training, so you could actually do it as well with after some software trainings that you performed.
Thank you, Svetlana. Elizabeth, do you want to add something?
I think most of it was already said. So yeah, you don't need programming skills. Of course, the more often you do it, the easier it is. But I also would like to mention that there's still Hamilton customer support. So something we also appreciate, that if you have a question, you can go back to the customer support and they assist you in finding a solution.
Okay, thank you. I think our next question is a little bit more into the scientific part. It's, does cfDNA give you any structural changes in DNA because it is fragmented as a result of cells undergoing apoptosis? This is for Elizabeth, I think.
Mm-hmm. I'm not 100% sure if I understand the question correctly, but so the main source of cfDNA is apoptosis and necrosis, but there are also other sources, as also active processes, for example. But typically it's from apoptosis, so you would see the apoptosis lettering. You often observe from dying cells based on the nucleosome. So on the length of the DNA wrapped around the histones, you would see that DNA lettering. And then you end up with fragmented DNA that you can work with, like with FFPE DNA, for example. I'm not sure if that answered the question.
For the person who has a question, you can always write down to say if you are for the clarification. We can all see here if you like for the qualification. If not, I would like to go to the next question, which is also scientific. What is the advantage between using a smaller panel sequencing approach versus a comprehensive whole genome sequencing?
Yeah, it depends on what you want to look at. You have to consider that a tumor cell is, or tumor is quite heterogeneous. So it could be that there's a certain clone, that there are certain part of the tumor which carries mutations that the other part doesn't. And that might be an explanation, for example, why the patient responds to a certain treatment or why he doesn't. Or for example, only in a certain part of the tumor, there are some resistance mutations which come up, and in the other it doesn't. And in order to see that, you would rather sequence a lot, so it would sequence with a high coverage. And that's of course easier if you focus on a panel, because otherwise it could be pretty expensive. And also, for example, when we use UMIs, unique molecular identifiers, then you would also go for a panel approach, because otherwise you would have to produce massive amount of data. But then again, if it's more exploratory and you want to get the full picture, then whole exome sequencing is rather the way to go. If you want to see new variants that maybe haven't been described yet, or you don't know what you're looking at, then whole exome sequencing is the method of choice.
Thank you, Elizabeth. Another question going back to automation is, what is the walkaway time and what is the hands-on time of the NGS solutions that you're presenting? I'm guessing that this has to do with the protocol in particular that you were presenting, Elizabeth, and also with NGS protocols in general. What is the walkaway time and the hands-on time?
I think I showed it in my slide. Then again, I'm not the automation expert. So there's still some hands-on time because you have to prepare everything, but it is reduced. I think it's like half of what the manual approach would be. So for us, for whole exome sequencing library prep, the hands-on time would be something like 3.5 hours compared to 6 hours of the manual approach.
And maybe I could also add something. So it's as Gabi, you said, actually, and it depends on the protocol. And so we mostly say just in loading the robot after you have prepared everything, like some of the reagents that you want to load on the deck. The loading takes maybe, Elizabeth, maybe you can also help me with like 10 to 15 minutes that you prepare all the plates, and then also all the tips that you want to load. And then it depends on the protocol and also on your throughputs, whether you have a so-called fully walk-away solution. If this works out where it's a rather short protocol, then you load everything, and at the end, the samples are ready. If you have also the on-deck thermocycle cycle included, and of course, except the QC control. And if you have a very high throughput and also very long protocol, then it might be that you might need also to reload some tips, but it just really depends on the protocol and the throughput.
Super, thank you. And we have another one, the scientific one is, could you explain to us? I have a little bit of problem reading these questions. Are needed for one sample running for CML. Who explains to us how many days are needed for one sample running, for example, CML? I'm not sure if, Elizabeth, you understand the question.
Well, typically for every library prep, it would take around 2 days or 1 or 2 days, depending on how you divide the protocol. For example, there are incubation steps, so usually it's a 2-day protocol. And then it depends on how you sequence it. For example, if you do a MySeq run, so how many samples you have and how much output you need. So if you then sequence the libraries, for example, on a MySeq, that's rather fast and you can have the data in the same day. For us, we mostly sequence on NovaSeqs, and there are also different flow cells available depending on the output you need and the amount of samples you have. And that could run, for example, for 2 days. So the actual sequencing then would take 2 days. So for us, really fast track. So from isolating DNA to library preparation, sequencing until the raw data, the fastest is between like 4 to 5 days.
Perfect, thank you. I'm gonna go with 2 more questions because I think we are a little bit beyond time, but as I said, feel free to ask more questions. The ones that are not answered right now, I will send you the answer after this webinar. So another question, or one of our last questions, is how small are cfDNA fragments? How do you prepare libraries from these fragments?
So typically you have a peak around like 160, 170 base pairs. So the smaller portion is like 150 to 220 base pairs. And then you have like multiples from this 160 base pair because of the apoptotic laddering, but you also have some larger fragments. But the most part, you usually have a peak in the profile around 160, 170 base pairs, so which is quite fragmented. But it's similar to working with, for example, FFPE DNA that from tumor tissue. And usually, so in general, every NGS process starts with fragmenting the DNA. So it's not such a problem that the DNA is fragmented. So we work with it just the way we would with FFPE DNA, for example.
Thank you, Elizabeth. And we have our last question is, are there scripts for Twist Biosciences NGS protocols available? This is from Svetlana.
Yeah. There is a list of protocols which is available, and I would actually advise you that you reach out to Gabi, and she will forward this question to the responsible territory manager, who then reaches out to you and will provide you the actual list of all NGS library prep methods that run on the NGS Star or also the other NGS solution. And one remark maybe on that, the list includes not only water scripts methods of the NGS library preparation protocol, but is mostly either validated by Hamilton and the users, like for example in this case together with CeGaT, or even by the vendor itself. So yeah, if you want to have, because this list is always changing and additional protocols are added up, you can just write an email to Gabi, and yeah, she will forward your request to the right responsible person.
Thank you,
But I can
Svetlana.
spoil that we are also using Twist, so yes.
Yeah, so the short answer is yes. There are. If you're just for the person interested, you can just write me an email with specific questions, what particular protocol you are interested in. If you want to know which ones we have, just feel free to write an email, and we come back with the answer. And with that, that was a large question. We need to wrap up. I want to thank Elizabeth and Svetlana for sharing their time, their experiences, and their insights. I also want to thank you who attended the webinar. I hope you found it interesting. And you receive tomorrow an email with a link to this recording. And with that, I want to say good evening, stay safe, and thank you for coming.
Thank you.
Thank you.
Bye-bye.